This Week in Parasitism is brought to you by the American Society for Microbiology at microbeworld.org/TWiP. This Week in Parasitism, episode number 85, recorded on March 9th, 2015. Hello, everyone. I'm Vincent Racaniello, and with me is Dickson Despommier.
Good morning, Vincent.
Good morning, Doctor.
Good morning, Doctor.
Really nice morning.
It's outstanding outside.
10:49 a.m. Did you know we lost an hour?
We did.
Also with us today, Daniel Griffin. Welcome, Daniel.
Oh, it's good to be here. And I feel like I lost 3 hours. I lost an hour with the time change and then 2 hours just trying to get here this morning.
Get here, that's right. That's right.
So I'm negative 3 hours.
Yeah.
The commuting has been difficult lately.
But the weather has taken a turn for the better.
I can't see behind me, Dickson. Is it blue sky?
It's outstanding.
You guys have
not only is it blue sky here, but it's nice and it's not balmy. I wouldn't say balmy is that's the wrong word
to use
at this time of the year.
The good view.
But
there's something new here today.
Right. We have...
Daniel has a laptop.
An anti-popper.
Yes.
I'm going to give everyone the view of what I'm looking at. And you have a cat climbing up down the back.
Isn't that kind of cute?
That's very cute.
Yeah.
I didn't see your little...
Should I spin it so that...
Would you please?
Look at that.
That's adorable.
Isn't that cute?
That is adorable.
Anyway, it's nice
How
weather.
Did you get that? You had that built into your machine, or
you buy it, Dickson?
It's special.
Buy it.
You and anyone
I would
else
like a
with
leaping
$3
trout.
or $4.
I want a leaping trout for mine.
6 degrees Celsius.
Right.
It's going to be. It's supposed to rain today. 65% chance.
That's okay.
No, we have enough water, Dickson.
I don't
know.
You can never have too much water unless it's flooding.
Yes.
We could.
My house is flooding. Always.
We could send it over to California.
We wish we could do that, right? They're building the wrong pipeline. They're pumping crude, dirty oil through it.
All right, let's do a show.
Hey, why not?
It is Monday morning. We forgot to tell everyone.
That's true. It is Monday
I'm a
morning.
little tired.
Are you now? Why is that, Vincent?
Mornings are rough in my home.
Yeah, I know. I
told my wife last
gathered.
night I didn't want to be a farmer.
No.
But I need to get
but
up.
You're living farmer's hours
getting
right
up
now.
at 5:15.
Not good.
Which is really 4:15 this morning.
This
right?
is true.
If I'd wanted to be a farmer, I would have applied for that job.
That's right.
But that's life.
But with me, you could be an indoor farmer and
my dad never complained. Of course, he didn't do a podcast.
Hours.
He would be out late doing emergency room
That's
things.
surgeons.
Come back at 4 a.m. and then get up and go out at 6. He never complained. He did fall asleep at the wheel though and run into trees all the time.
Oh, really?
He did it 3 times.
Oh my goodness.
Yeah.
No,
I laughed about it.
Oh, but that's not to be laughed at.
And he would pick me up from college with a new car. I said, "What happened?" He would laugh.
Oh dear.
He said, "I fell asleep and ran into a tree." Or once he ran up on someone's lawn.
Wow.
Another time he ran into the guardrail.
Oh, Vince.
Turned out he used to take this blood pressure medication called Inderol. Remember that?
Yes, I do.
And it made him sleepy.
Yeah.
Oh my goodness.
It will do that.
That coupled with his life. But he never complained. I should learn from my dad. He never complained.
How about the people he hit? Did they complain?
No, the insurance took care of it.
Yeah, right.
He fortunately bought big cars because back then, that's what you had.
Wow.
He'd walk into the showroom and say, "Give me a car. I need a car."
Wow. The biggest one.
He'd write a check. Boom.
Amazing.
That was it.
Amazing.
Oh well.
Interesting.
Anyway, I have to learn from my parents. They never complained. And I complain all the time. And I use my podcast as a forum to complain.
Right.
We had a lot of responses to TWiP 84 case. And by the way, we continued to receive answers to TWiP 83, and I've grouped those with the regular email. We can get to those later.
Okay, great.
But TWiP 84, do you want to... I can summarize this one. I remember it. But you better do it.
All three of us.
So this was a gentleman, a young man that you saw.
So this was, as our listeners may remember, a 32-year-old young man who likes to drink a bit.
Hang out with his
pals. A bit much at times with his friends.
Yeah.
And he had presented with 3 months of recurrent fevers, headaches, productive cough, and he is from Missouri. And as we mentioned, he does the things there that kids his age do, such as drinking a little too much, tubing down the rivers. There was the history of a raw crawfish consumption.
Is it crawfish or crayfish?
Depends on where you're from.
So from Dickson, it would be craw.
Craw, if you're
You
from
say
New
craw?
Orleans.
You say craw?
Crawfish. No, I say crayfish because as a fisher person, I wanted to degenderize that one. You can use crayfish as bait to catch smallmouth and largemouth bass, mostly smallmouth bass. And in that state of Missouri, it is like smallmouth bass capital of the world because all of the rivers there drain through deposits of limestone. And limestone neutralizes the rivers and mobilizes the calcium and allows for large populations of crustaceans. And this particular critter, the crayfish, happens to be a crustacean.
When I was a young boy, we had streams around our land.
Yes.
Yeah, or I would find them, and I always loved when you could see some life on the bottom.
Exactly.
A crayfish crawling along,
That's right. Yeah.
Or a little fish.
That's right.
All right, we had lots of responses. Jan wrote, this is fun, and although I'm sure someone will gripe about Dickson's enthusiastic response to the crayfish, it made my life easier. I think it's Paragonymous calicati. As for eating raw crayfish, how drunk would you have to be? Right. Jan is from the Netherlands. P.S. I'm not an expert in Dutch elm disease. I'm a tree worker. Inspecting trees for potential dangers is part of the job, so I have a working knowledge of the most common problems. Dutch elm disease is caused by a fungus, Ophiostoma, and spread by beetles.
Look at that.
PPS, the weather in Rotterdam. Heavy showers, 7C, winds up to 6.
That's why they call it Rotterdam.
Robin writes, thanks to Larry Page, Sergey Brin et al., and to Steve Jobs, Steve Wozniak et al., like shooting fish in a barrel, Paragonymous.
And even, wait, the other one actually gave it a species name of calicati.
Yeah. How would you know that?
Well, there are many species of Paragonymous out there. Westermani is another one. But calicati has an unusual history of discovery. I don't know if any of you in this audience today, sitting around this table, know that history.
No, I don't. But
I would
others
love to hear it. I
others
love
have
history.
Sure you would. It was discovered in a Bengal tiger in a zoo.
Calicati?
Yeah.
He had eaten a crawfish?
Well, where they lived originally, it had. They're water-loving animals. The
Tigers? Yeah.
Tigers love water. Yeah, one of the few cats that actually
By the
does.
way,
Jaguar is another one.
I'm reading these in the order of receipt.
Aha.
Chris writes, just as I was about to send you my answer of lymphatic filariasis, the new TWiP arrived, and I was thwarted by your rapid turnaround. I'm loving TWiP 2.0. There you go.
Good deal.
And have been trying to infect others with it.
Good.
I've been listening since TWiP started and encouraged me to go back to uni and become a medical laboratory scientist as a mature-age student.
Nice.
Since graduation, I've ended up in cytogenetics, but still look forward to each episode of the TWi Triumvirate. Weather today in Brisbane, Australia, 30°C, 60% humidity, and little wind, so a lovely autumn day. Today's case did intrigue me somewhat, particularly with the intermittent fevers and lack of travel. The key, however, is consumption of raw crustaceans, Paragonymous species, most likely calicati in the U.S. Metacercariae insisted within the crustacean, exist in the duodenum, penetrate the intestinal wall into the peritoneal cavity, then through the abdominal wall and diaphragm into the lungs, where they become encapsulated and develop into adults. The worms
Isn't that an unusual route of infection?
reach other organs and in tissues, including the brain and striated muscle.
Yeah.
The patient presented during the chronic phase, during which pulmonary manifestations include cough, expectoration of discolored sputum, hemoptysis, and chest radiographic abnormalities. Extrapulmonary locations of the adult worms result in more severe manifestations, especially when the brain is involved. Hey, what's hemoptysis, Daniel?
So the heme being blood and the
blood in the sputum,
and then coughing it up. So it's coughing up of blood.
Hey, do you know what? What's that word we learned on TWiV? Let's just go.
You learned it very well.
I
learned
a lot.
I'm old.
Problem is, I learn them and then I forget them.
I'm old. Sternutate.
Oh, what is
this
word?
Sneezing, sternutating.
Oh,
Snerutate?
Our pediatrician friend in Cambridge sent it to us.
So it'd be sternatus or something. Was that when you sneeze up blood?
Diagnosis at this stage can be by the presence of eggs in the stool or sputum. Concentration may be required. Immunoassays are matched to Paragonymous westermani. You may not pick up calicati. The 3-month delay in seeking treatment gives sufficient time for eggs to become present for diagnosis. Treatment with praziquantel is indicated with corticosteroids to reduce inflammation caused by dying worms. Now, Elise is the young lady down in Lower Manhattan who was very excited to get the first one right. I was so excited to have heard my letter read on TWiP and to have gotten the diagnosis right. I had to plunge in again.
Sure.
And I suppose that is an apt enough metaphor given the case study of the week. I believe the 32-year-old gentleman is suffering from paragonomyosis, infestation of lung flukes. So she goes on to talk about eating the crayfish.
Yeah.
And the symptoms which we heard about, fever, cough, exhaustion.
This particular parasite, I'm sure we're going to get into some details, but I just want to interject here that she mentioned the lung fluke.
Yeah.
Because typically it does locate to the lungs, but that's not the only place it can go.
Right. I do have a question about this patient's fevers, which had come and gone for 3 months, which is what causes the pattern? Is it linked to the eggs being released in the lungs, they're hatching, or some other part of their life cycle?
Good question.
Another question, why these cases started coming up more in 2010. Most of the sources I found, not just the Float, Missouri folks, even there is a website she cites called Float, Missouri.
Yes.
Warning floaters, people who float recreationally, not to eat live, raw, or even dead crayfish. I skipped that over. So how did this parasite arrive in large enough quantities to be noticeable? I fear the question is about the arrival of the fluke, since there is zero chance that eating raw crayfish while drunk on the river is a brand new diversion. I hope I'm right. These exercises are an absolute pleasure. Lower Manhattan is getting showered with little ice pellets now.
Yes.
That was last week.
Yes.
Dr. Wink, as an ID physician, I'm used to being nonplussed or not judgmental by my patients' histories. However, as a vegetarian, I was repulsed by your most recent TWiP case presentation, paragonomyosis. TWiP Duo was always great. The TWiP Trio is fantastic.
Oh, good.
And he sends a MMWR article warning.
Oh, that's great. That's a great publication.
Dan writes, case under discussion, dealing with the 32-year-old male with peripheral eosinophilia fever and symptoms of lung infection is Paragonomus calicoti. My diagnosis is based entirely on a peek at the literature and a paper which he sends to. And then he diverges. Should we diverge? I'd like to mention another point that I've been meaning to address since it was incorrectly stated in the podcast by Dickson.
Again.
Hibernating bears do not have circulating antifreeze proteins. We find these in teleost fish sera in Arctic waters of the North Atlantic and elsewhere in other organisms, but not in hibernating mammals. I studied these proteins in the 1990s, and he goes on to tell us what he did. He published a paper.
You're going to have to tell me why Trichonella survives in their muscle tissue.
Well, how do you know that it has anything to do with... He's saying they're not, so you better believe him. I cannot resist.
Hang on. You can freeze bear meat and it doesn't kill the larvae.
Hey,
whereas if you
Daniel,
freeze pig meat...
You can hit
it kills
Daniel.
the larvae because you get ice crystal formation. So you're going to have to account for
there's
the
some
difference.
other reason. There's some other reason.
It's got to be... No, it has to...
AFPs function to alter the hydrogen bonding patterns of water such that the isodiametric nucleation of the ice crystal is thwarted by the alpha helical secondary structure. Bears do not need to thermoregulate like fish. Rather, they live in dens that are well above freezing, and indeed their body temperature never drops more than a few degrees. That's not what I'm saying,
though. You kill a bear and you freeze the meat, and it doesn't kill the larvae. Forget about hibernation. It's freezing the meat.
Let me finish. I could explain the lipid metabolic pathways by which hibernating bears convert depot fat to NADH, etc. And then he goes on to talk about... glycolysis. Okay, so the final question you might have would be the following. Why don't bears in hibernation suffer from ketoacidosis? I can explain this, but it takes too much space on this email. If you want to know, let me know or try to tease it out of the literature. Right. So, no, bears don't have AFPs in their muscle. That is rubbish. So you are rubbish, Dickson.
Okay, well,
also, they are not enriched in other forms of paradigmatic frost tolerance mechanisms, such as high elevations of carbs or sugar alcohols. I should make another comment that is always in my mind when I listen to podcasts and increasingly when I read biomed literature.
Uh-oh.
He's a teacher. Biochemistry is being increasingly avoided or diluted in the medical sciences. There are fewer and fewer investigators who know their biochemistry and molecular genetics. Because of this, I repeatedly encounter mistakes in reasoning and accuracy when biochemistry is brought to the front. This is radically obvious when people stumble over lipid metabolism. Too bad, since we have an obesity epidemic. Finally, the rounding out of the host with Daniel is a welcome event. Keep him on board if you can. Ciao.
If we can. We pay him top dollar to come here.
Yes, I heard they doubled my zero salary.
We doubled it. We will triple it. Come on, we're going to steal you away from anybody else.
Varun from India writes, he's from Bangalore.
Yeah.
I would like to have a BAL done, bronchial alveolar lavage, done for parasite eggs. High eosinophil count rules out bacterial and viral etiologies.
I don't know if that's true, but I would probably say the highest eosinophil count is more consistent with the parasitic etiology.
Or metazoan infestations.
He guesses Paragonymous westermani.
The westermani is an Asian
and he says
species.
Maybe P. calicati, treat with praziquantel.
Right.
The TWiP reboot keeps getting better and better. Thank you for the time spent in educating.
Great.
Trudy is our friend from Naples, Florida.
Nice.
Thank you so much for the reboot. Love the case of the week and the mental stimulation it provides. The unfortunate crayfish cruncher had paragonymous or lung fluke. I'm a former public health nurse epidemiologist. I suspected a fluke almost right away. In public health, epidemiologists case classify and don't diagnose unless a qualifying medical license is held. I used an ASM journal via the Search Medica app as well as a search via Netscape, which resulted in a link to this article, P. calicati flukes in Missouri. Okay. And then she says the treatment, etc. The biggest hint to me was ingestion of raw pond shellfish.
I mean, you have to give us some kind of a hint, right? Otherwise, we're shooting in the dark here.
And John writes, I love the new TWiP format. Both the case studies and the more frequent episodes, keep them coming. I particularly enjoyed Bigfoot. Your discussion of tsetse fly paper made me laugh out loud. Fortunately, it's spring break here at Creighton University, so no one was around to hear me. I enjoyed your discussion of the case study, which includes several engaging factoids and anecdotes. I'm currently teaching zoology, and we recently covered Wuchereria. Wuchereria.
Look at him.
Well done.
Whoa, he's come up to speed really quick.
Next time I teach it, I'll be sure to include some of these stories. Students remember the material better when it's accompanied by interesting narratives.
Yeah, that's true.
The great thing about parasitology is that it's more than biology. It touches on so many different subjects and disciplines. To paraphrase your car talk counterparts, parasitology is historical, folklorical, economical, pharmacological, socio-political, and pathological. Is that what they used to say?
All of the above, everything.
Then he guessed Paragonymous calicati, common fluke in the Midwest.
Yeah.
Ken, episode 85, Bigfoot, another good episode. I'm looking forward to the case studies. And he guesses Paragonymous. Dickson's leading questioning of Daniel and focusing on crayfish pointed me to it. Sends us an article.
Guilty as charged.
Keith guessed Paragonymous calicati.
Look at this.
Sorry, very short. I love the
exam.
cases. I love TWiP. The addition of cases every week is fantastic. Keep the TWiP and the case of the week coming. Although this email is short, at least you know people are listening and enjoying. It doesn't matter if you're short.
Oh, that's exactly
Thank you,
right.
Keith. So there you go.
Yeah.
Daniel, everyone got it.
Did they? Yes, they did.
Well, I shouldn't have said that.
No, you were right.
That's right. You haven't revealed yet.
I haven't revealed it.
So
but
tell
I
us
think
how you figured it out.
Well, in this case, I saw one of our letter writers here requested a BAL, a bronchialveolar lavage, and that's what this patient underwent. And I'm going to show this picture to our parasitologist
on call
with lab experience.
Yes.
And I'm going to describe, as he's looking at this, what he's looking at.
Yeah.
So eggs were recovered on wet mount.
Yes.
And these eggs were about 100 microns long by about 50, 60 microns wide.
Yep.
They were yellow-brown,
Yep.
oval-shaped.
Yep.
They had a thick shell. They were asymmetrical. One end was slightly flattened.
Yep.
And that end is what we call an operculum, which is something that can open, an opening. Right. And then the opposite end is somewhat thickened.
Right.
And then next to it, something we didn't actually see on the BAL, but something that looked like a flat red leaf. It was a leaf-looking thing.
Well, yeah. If you look at that flat red leaf, that turns out to be the adult.
Yes.
You actually lavaged up an adult?
No.
I didn't think so.
The eggs were what came out.
That's what I thought. So the egg itself is a very typical egg for this parasite. It's hard to miss it, actually. And the operculum is like a little manhole cover. It's circular, And when the organism inside, which is referred to as the miracidium, it's a ciliated, free-swimming form of this life cycle stages. We discussed that before with the schistosomes, which are also trematodes. So almost all, in fact, I think all trematodes have a miracidium stage. And this actually seeks out the next victim on the long chain of events that leads to this infection. It's looking for a snail. So, and it finds it by chemical trails that the snails leave behind in the water. And this miracidium then finds the foot, usually of the snail, which is a soft tissue, and attaches and penetrates through and then enters into the hepatopancreas of the snail, because it's a combined organ. And it starts to undergo some remarkable changes, which results in the multiplication of this parasite within the snail itself. And then from that intermediate host, we get the emergence of the Sicariae. And these Sicariae then, in some cases, insist on inanimate objects, or at least not animal life forms, like, for instance, the case of Fasciola hepatica, one of the most commonly distributed flukes, also known as the liver fluke. You get this by eating contaminated watercress at the edge of a pond that had sheep. But in this case, these Sicaria seek out crustaceans.
Really?
Freshwater crustaceans. They don't attach to anything other than freshwater crustaceans.
They attach to them.
They attach on the surface. They don't go inside of them. They just insist on the outside.
Now, how does this guy get the infection?
He had to put the whole crayfish in his
squeeze out
mouth
the guts,
to get out. No, not the guts. He was trying for the tail meat.
Yeah.
Which
but
is
in the meanwhile, he got the...
Yeah, so they came off in the process. And that's what happens out in the...
it's not actually from the meat itself.
No, it's not from the meat itself.
So if he had taken the meat out carefully...
Yes, he would have been fine.
And eaten
or if he had just ripped off the tail. So if you're drinking too much, floating down the Missouri, and you just rip off the tails, then you're going to... then he would be okay.
Let's put all that together. No. You still have to squeeze the meat out from the tail.
He was showing off.
That's
he
right.
put the whole thing in his mouth.
Yeah.
Hey, guys, look at that.
That's right. Something like that. But in Asia, they're not showing off. They are eating raw crab, or something called drunken crab.
Yeah.
Which is submerged into some alcoholic solution to slow them down, I guess, so they can manage them. And then they crack them open with their teeth and pull out the meat and eat the meat. And that's how they acquire this infection.
So are there any other shellfish that I should be aware of? So sometimes in a restaurant you get cooked shellfish, but that's not a problem because it's cooked, right?
Name a shellfish that you might be worried about.
Stone crab, I'm thinking.
That's a saltwater variety.
Saltwater, so it doesn't matter.
What else?
Besides crayfish, what other crustaceans would we eat from freshwater?
Well, there are freshwater prawns and shrimp that grow, not in this country, but in Asia. They have a lot of examples of that, and lots of freshwater crabs also.
But the saltwater things are all boiled anyway, right?
Right. So I was in Shanghai not too long ago, and there's a hairy crab season.
Hairy
my
crab.
wife and I looked at each other and said, what are we doing here? And then they served them. They were actually delicious, but they were very unusual looking because they had lots of hair-like projections off of their legs that caused them to look hairy, and it was a delicacy.
Daniel, what did you tell this patient after you diagnosed it?
So, this is great. All we have to do is go through the emails, and they tell us how to treat it. This patient initially was pretty sick and ended up getting 3 days of praziquantel, and eosinophils came down, fevers went away, and follow up a month later, the guy was doing great.
Did you tell him?
And of course, he was encouraged to not drink excessively and eat raw crayfish.
You told him that was how he acquired the infection.
That is how he acquired.
Was he surprised to hear that he could get infected from a crayfish?
Well, I always have an ulterior motive when I throw one of these cases, and one of them is to let people know that there's parasites right here. You don't have to travel. And actually, the case we'll present later is along these themes too. Is we always think about parasites as other people's problems, right? These drunken crabs and somewhere in the Far East. But here's just a guy floating down a river having a good time, and next thing you know, he's almost going to die from a parasite in his lungs.
Dickson, if we went to your fishing hole in Pennsylvania, would we find crayfish in there?
You bet.
Could we find one of these parasites?
If we looked hard, we might be able to.
Because
there are other animals, there are other mammals that can catch these. things.
Who would eat a crayfish? A bear?
Raccoons.
Raccoon. So you would find these parasites in raccoons?
I believe so. It's got a very promiscuous host range of activity. Remember, I told you that Pleurogonomus calicati was originally found in a tiger, but we can catch it also. So it infects a lot of varieties of animals.
If we took crayfish out and looked at them, would we see the?
No, they're microscopic.
Small, right?
These Metasicaria. Yeah. Actually, if you got out my textbook, you could see a picture of one because I did photograph, although they're hard to get. A photomicrograph, I should say, of Paragonomus metasicaria. And it's got a little bitty, it looks almost like the adult parasite inside, all wrapped up, in this little cyst. And that's actually what's going to happen as soon as you ingest this thing.
But where would it be in the?
It would be under the trematodes.
Trematodes, Paragonomus westermani.
See, there it is.
But you don't have Killicati.
We don't need Killicati because we're not really interested in one or two cases a year in the entire country. Yeah, there it is. That round discoid shaped.
And you have a little life cycle here.
Well, not a little life; it's a big life cycle. Actually.
You have
a
whole
page.
You have
a whole page
a
on that.
Tiger here.
We do, and pigs too. Notice.
And a crab.
That's right. That's right.
This is a crab, but it's not you actually. It's a real crab.
That's a real crab. I just crab a lot. But we used to have one of those as a preserved specimen, just to emphasize to the students that this thing is a very unusual parasite. But if you go to certain places, crabs are on the menu almost every day in almost all the restaurants.
There's a lung cyst.
Right. So an unusual feature of this infection is as follows. We've talked about this before. Mm-hmm. There are environmental clues that this worm uses because they're hermaphroditic. Okay, they have male and female organs inside, but they don't self-fertilize. So it needs two parasites in order to complete this life cycle. So if he was lucky enough to eat a crayfish that only had one Metasicaria on it, and it turned out to be it's both a male and a female, he might have had hemoptysis from a single infection, but he would have never been producing eggs, so you would have never detected it. Mm-hmm. You might have had to biopsy to find out or even operate, thinking this might be some kind of ulcerative cancerous lesion of some sort, but it wasn't.
Could this have been lethal?
Oh, yes.
Untreated.
It has. Yeah, when you accumulate many of them, you can die. Sometimes they can locate to the brain.
That's, and there have been cases, unfortunately, of that where they go. And I think, again, our emails are great. One of the people points this out. We used to have this idea with a lot of parasites that, it's the lung fluke, it goes to the lung.
Right. Or the liver fluke, it goes to the liver.
Yeah. Or I think at one point we'll present a case of neurocystosarcosis. It goes to the brain. But what we're beginning to realize is they go everywhere.
They do.
When they end up in a host, particularly that they didn't necessarily evolve to be in, then they maybe wander even more.
Sure. We would call that an aberrant infection.
Yeah. So in a case where, and this person actually fortunately did not have any findings to suggest CNS involvement, but if they do, you'll often add a steroid to the treatment to avoid some of the inflammation that might trigger seizures and syphilitic reaction. Sure. But yes.
So I knew someone who worked on this parasite. His last name was Sogandaris Bernal. I'm sorry. His first name was Sogandaris. His last name was Bernal.
Was he Bernal?
No, he was a very nice man. And he studied it, I think it was at LSU, as I recall. And he worked on the life cycle. And what he would do, you can infect a lot of different kinds of animals with this parasite. What he would do is he would infect a host with one Metasicaria. Mm-hmm. And then several weeks later, remember, you need two in order to complete the life cycle. He'd infect it with another one. Now, imagine how big the lung fields are on a pig, for instance. Big, right? Just like ours. Mm-hmm, mm-hmm. So here's this parasite, one, localized to one of the lobes of the right or left lung. And then he infects with the next Metasicaria, which hatches out into an adult or a micro-adult in the small intestine. And then it migrates through the intestinal wall, through the diaphragm, into the pleural cavity, through the capsule of the lung, into the lung, and finds the other parasite.
Finds it, huh?
Finds it.
You must be chemical trails, right?
Yeah, but what kind of a chemical trail are you going to pick up when you penetrate the peritoneal cavity? I mean...
Penetrate the peritoneal cavity. Wow, that's an alliteration, isn't it?
Yes, but...
Well,
Paragonomus penetrated the peritoneal cavity.
You only need molecules, Dickson.
Yeah, I know that, but how do those molecules from that worm in the lung get out?
Circulation.
and then creating a gradient
We do have a circulation.
that this parasite can follow. What if it enters the wrong lobe?
It'll eventually get there. It diffuses.
But it's in the other lung.
Yeah, it's okay. I don't have a problem.
No, I have a big problem. I don't have a problem. I have an issue with that. That's a miracle that they find each other like that. So I used to give this lecture about schistosomes to the students here.
They find each other delightful.
Okay, fine. You just stepped on my line. That's fine.
Yeah, but it is a great.
That's what you were going to say?
That's exactly what I was going to say.
It was the name of one of our episodes.
I know it was, because I just wanted to refresh everyone's memory to the fact that these parasites follow environmental cues. And they don't care where they locate as long as they find each other.
Yeah. But it's a great life cycle, and the flukes
Fabulous.
are maybe, I say, sort of one step more complicated than a lot of our parasites as far as the life cycle. So
that's true.
As Dickson is saying, they start off in the intestines, right? And ultimately, they kind of want to get back out, and so some of them are going to get passed out through the intestines. Some of them will be getting coughed up, so they can get back in the environment. But it is really interesting, and at some point, I think you discussed before, we'll discuss again the life cycle of Ascaris, where, again, things like to, and in that case, they don't even really want to stay in the lungs. They sort of pass just through, so they get coughed up, swallowed, and get back into the intestine.
Exactly.
And, but then it goes out, and almost all these flukes have this snail intermediate.
All of them. I don't think there's an exception.
Yeah, I don't think there's any well-known human. I think somebody once wrote there is one fluke out there that uses something. I think it's in one of your past episodes, but it's not one of. I will say not one of medical importance. But yeah, in general, flukes, except for this rare exception, that. And maybe that person will write in again and remind us.
Sure, they will.
Or our listeners can go back and re-listen to all our prior podcasts.
Oh,
but
that's torture. I would never want to do that.
But then the
Dickson.
tricky part that's hard to remember is after the snail, where does it go? Is it going to end up in watercress? Is it going to end up in a crab? Is it going to end up, in some other form of crustacean?
Oh, yeah.
And trying to remember that is always been the challenge.
Or a fish, the cyprinoid fish in Asia. The Metacicaria often locates underneath the scales of the fish.
Yes.
So you'd wonder, how can someone get infected when it's on the outside of the skin, under the scales? How would they,
Dickson?
Well, they eat the whole thing.
Mm-hmm.
Dixon, you can't, if you produce something like a podcast.
Vincent, you're taking offense when I'm joking.
You can't tell people it's torture to listen to it.
It was a joke, Vincent.
Who knows when you're joking?
It was a joke.
No, I don't think so.
Actually, one of our email that came in that talked about how when he listened to the Bigfoot episode, he actually laughed out loud. I listen to our podcasts after the fact, and sometimes find listening to them after the fact even more enjoyable than being here live.
Oh, it is. It's very different.
And so, I was listening to it on the A train, and when you laugh out loud on the A train, because you've got your earbuds in and nobody knows why you're suddenly laughing,
that's right,
you get extra space.
Yeah.
It used to be that they would throw a net over you, but they don't do that anymore because everybody's got earbuds.
All right, let's move to our paper, Dickson. What do you think, Daniel?
Yes,
but as a coda, as a little coda between the episodes.
Yes.
I'm looking at Daniel's laptop with the cat hanging down. Do you know they make a sticker with Snow White holding the apple?
Yes.
Really?
Yes, because it's got a little bite out of it, Dickson. You see on the back of mine?
Is that the Wicked Witch or is that Snow White?
Snow White.
Oh, okay.
And she has her hand under the apple. And it's wonderful. It's just wonderful.
How interesting.
All right. We have a paper today that was just published in PNAS,
which stands for the Proceedings from the National Academy of Sciences.
That's right. What is the Proceedings, Dickson?
These are the highest level articles ever published in the United States, according to the PNAS.
What is the National Academy of Sciences?
Well, it's an august body of accomplished scientists.
How do you get in?
You have to be nominated by someone who's already there.
Neither of us are in the academy.
Have you ever been nominated?
I have been.
So have I.
And we're just failures.
We just missed.
We
missed by one vote. I know.
I wouldn't.
See, that was another joke.
And you
didn't even know I was joking then.
I wouldn't want to be in any club that would have.
That's Groucho Marx who used to say that.
Who is
No, Woody. Isn't that Woody Allen?
No, Groucho Marx.
Groucho Marx?
Said that. I would never become a member of a club that would accept me as a member. Knowing that their standards are so low, I wouldn't want to be a member. But I would, if I was asked to join the National Academy of Sciences, I would do that.
Yeah, well,
yeah, sure.
You just wait.
That was not a plug for. Go ahead and
wait for that phone call.
You can't be renominated.
It could be that your TWiP, your work on
if you get nominated and don't get in, you're out. It's all
I
over.
believe
Is that true?
But they have 10 years to make up
you
their
got a
mind.
long time.
Yeah.
It could still happen. But
No, I exceeded that limit. I think I'm gone.
Our colleague Steve Goff is in,
He is.
but our colleague Saul Silverstein is not, which is really a shame. He
That's
should be in.
Odd.
The problem is that it's a highly politicized organization.
Are you saying that, Vincent? Are you not joking now?
I'm not
You're just
joking.
not joking.
And people will not vote for others who they don't personally
Can you-
like, even though their
Can
science
you mention
is
an
wonderful.
organization by election that's not politicized? I would like to know one because I don't think that's in the human
Perhaps
genome.
I used the wrong word.
It's biased.
It's biased. The election procedures are flawed. Enough of that. The paper is called Human Trypanolytic Factor ApoL1 forms pH-gated cation selective channels in planar lipid bilayers. Relevance to trypanosome
That's
lysis.
a mouthful.
It is a mouthful, and we are going to take it apart.
Great.
Go ahead, Dickson.
Okay.
You know why I picked this?
You didn't, actually. I thought it was Amy that picked this.
Yes, Amy in my lab sent it to me, but I picked it
Oh, okay.
because
Why did you pick it?
ApoL1, apolipoprotein number 1,
Yes.
is one of 6 different proteins
Yes.
in humans.
Yes.
So it's a gene that's been duplicated 6 times,
Right.
and apolipoproteins are those things in your blood that carry cholesterol, right, Daniel?
Yes.
And this-
Transporter molecules.
Yeah, actually, this is part of HDL, part of
That's
the
right.
HDL complex.
Right.
And a few years ago, we found that ApoL, which number?
Yes.
ApoL6 can inactivate poliovirus.
Get out of here. Really?
All right, I'm going.
I'll be darned. No, please stay. I know the person who discovered this, by the way. This phenomenon that we'll be discussing today, I knew this person very well. As a matter
So
of
anyway,
fact.
it's interesting that these have a role in lipid metabolism. Just the Daniel, our letter, we don't
Right.
know a lot about lipid metabolism, but we
No,
know.
I'm actually my
finding out
appointment
now.
is in the Department of Biochemistry. So apparently if there's biochemistry questions,
well, then
I'm
you-
supposed to know the answer.
Anyway, so it's interesting that 2 of these are antimicrobial.
That's
Isn't it?
remarkable.
And maybe the others are
It
as
is
well.
remarkable.
So the way we found this is we have a plasmid encoding ApoL6. Okay?
Yes.
We put that in cells to overproduce the protein.
Go on.
We infect with polio, and it reduces the yield.
Reduces.
It reduces.
Doesn't eliminate, though. It
Ooh,
reduces.
Which reminds me, I have a progress report due in 6 days.
That's important.
I better get going.
Right. So I know
Um,
the person who
yeah,
they
it
referred
reduces.
to originally on this. 1978 was when this was discovered.
So what's the relevance to trypanosomes, Dickson?
Well, Vincent, this is...
If you can tell me if you don't know.
No, I can. I know the relevance to it because it allows humans to resist becoming infected with almost all of the species of trypanosomes, except, of course,
So that
the one
includes...
that causes major disease in West and East Africa.
Tell us the species.
This is Trypanosoma Brucia, first of all. So
That's attacked by this.
it is, and it's killed.
So-
and then
and what
there's
else?
Trypanosoma evansi. That would be attacked and killed too. But that's transmitted from horse to horse by sexual contact. So we wouldn't encounter that parasite.
But
I'm
which
sure.
are the ones that we do encounter?
Right, except maybe, Caligula. Who, forget about that.
Yeah, please.
That was a bad reference.
We can edit him out, right? When
We
he's
can, but
inappropriate,
there
can't we?
are numbers of... No, I wouldn't do that. When I
We have
would leave-
we have children who listen.
Remember Caleb? No, Caleb doesn't listen to Trip.
Trip.
Tripping trips. So that's what we're going to call this one, tripping trips. But there are 2 species of trypanosomes in Africa, which are transmitted by tsetse flies.
Yeah, tsetse.
True.
I noticed you said tsetse.
Tsetse. I say tsetse. Some people say tsetse flies, but I say tsetse flies.
Mm-hmm.
And in East Africa, we've discussed these at length in the shows on them, of course. There are multiple reservoir hosts for Trypanosoma rhodesiense,
Rhodesiense. Okay.
which is found in Rhodesia, but Rhodesia doesn't exist anymore. So we think about changing the name to Zimbabweanse, which is what Rhodesia...
Is that really being considered?
No, of course not. That was just meant as a throwaway statement here, because no one knows where Rhodesia is anymore because it doesn't exist.
Well, when people die, do we change the organisms named after them, Dickson?
Sometimes we change the names of these buildings here. You notice we have a complex of buildings. You can get it changed if you donate more money.
Wucheria.
Wuchereria.
Wuchereria.
Wucherer.
Wucher and Bancroft.
Bancroft, that's right.
They're both dead.
They are.
But we didn't change the name of the organism.
Did not, but they changed the name of Pasteurella pestis.
For other reasons, though, not because Pasteur is
It doesn't matter
dead.
why they changed them. It matters that they changed
So
them.
these two, T. rhodesiense, what's the other one that infect human?
Gambians.
Okay, they infect, but they're not affected by this trypanolytic factor, right?
Except for a few mutations that some people
All right.
have, but
We'll get to that.
recent. Yeah.
So basically, there's something in your blood. It's called trypanolytic factor, which is a high molecular weight complex that will lyse these two trypanosomes.
That's correct. And with the trypanosomes...
Sorry, Brucia.
Well, it'll lyse all the trypanosomes except the two that affect people.
And they contain
the
a
East and West
substance,
African.
SRA, which is abbreviated SRA, which counteracts the effects of apolipoprotein 1.
The trypanosomes have SRA.
Yes.
Which stands for what?
Take it,
Oh, I always
biochemist.
make notes because I can never remember all these things.
So that stands for...
Serum resistance associated protein.
Right.
Yes.
So
I had to make a note. I couldn't remember that. Can you imagine that?
now,
it doesn't stand for anything specific.
ApoL1
Yes.
is part of this trypanolytic factor, but it seems to
is.
be the active part, right?
That's right.
And do you know what the really good evidence for that is, Dickson?
Well, you can incubate trypanosomes in it and they'll lyse.
Right. And would you like an in vivo correlate of that?
Tell me.
Well, you make transgenic mice with the human gene
Look at
encoding
you.
ApoL1,
Yep.
Yes.
and then you can infect them with
Trypanosoma
Brucii,
Brucii.
and it will prevent infection. So that's
Right.
pretty nice. It shows you that this
Yes.
ApoL1 is very
It has
important.
relevance to biology. That's
So the SRA is something produced by the trypanosome
yes, as a counterattack.
that counters this
That's
ApoL1.
Right. That's right.
And now you said that in parts of Africa, the humans have mutations in their ApoL1.
These are recent. These are not Africans living in Africa. These are African Americans living in the United States. And they have found that some people living in the United States of African origin, but now living in the United States, contain a mutation in their apolipoprotein 1, which negates the effects of SRA
Right.
and allows them to lyse the trypanosomes that would ordinarily infect them and make them sick in Africa.
But what's
there's
the
a price to
problem?
pay for everything, of course, and in this case, there's an association with increased glomerular nephritis, I believe, where you get actual disease of the kidneys based on this mutational event, which allows them to resist African trypanosomes, but of course we don't have any in this country, so that's not applicable in this case. There's no natural selection for them here. And I don't even know if there are in Africa. Dan, have you heard anything about whether there are subpopulations of humans living in Africa that are resistant?
There are actually, and they
Okay,
discuss
because you spent time in Africa, right?
Yes. They discuss in several of these variants, this G1 and G2 variant.
right.
And as we get in the mechanism, maybe it'll make more sense what's going on here. But this is similar to, let's say, our G6PD deficiency or our sickle, or where if you're a heterozygote, it's perfect. So what you're doing is you're producing one of these that's going to not be neutralized, and that's great.
Yeah.
But when you produce two, and it's being produced in the glomeruli of your kidneys, then unfortunately the consequence is kidney damage. And I ran across this first in the context of accelerated kidney damage, this ApoL1, before I understood its role in the trypanosomes. And it made it to the New England Journal for that. And as Dickson points out, this probably only arose about 10,000 years ago, probably under selective pressure in the African, probably sub-Saharan region, because particularly Western trypanosomiasis is such a huge issue in the human population. And I think it's gorillas, green monkeys, and humans are the only ones that have developed this ability to basically lyse the trypanosomes.
Not all such alleles are damaging to people. I can think of at least one that protects against an infection, but has no apparent consequence, and is a deletion of CCR5.
Yes.
Which makes you resistant to HIV infection, but doesn't seem to have any clinical consequence. We really don't know what the selection pressure for that was. It's a mystery, but about, I think, 5% or so.
Or even what the CCR5 does then.
Well, it's a chemokine receptor. We know what it does, but apparently it's redundant.
Maybe there are alternatives.
Yeah, exactly.
Yeah.
But about 5% of the population has this allele.
That's very interesting.
And I think loss of Duffy put it in a malaria kind. I'm not sure there's
Right.
any negative consequence to losing Duffy,
Right.
which was something that allowed.
Well, because there are so many other blood group factors, right?
Mhm.
That seem to be. I don't even understand the reason why we have an ABO system.
Yeah,
What does it mean?
I don't know.
I have no clue.
Does it have to mean something?
Well, it means that you can't donate blood if you're
Yeah.
It's harder
you
to
can
get
get away
harder to get transfused.
exactly
because of
right,
all
but
those problems.
I'm not sure what the biological significance is.
So in this paper, they're trying to figure out what ApoL1 is actually doing, right?
Right.
And there is there's been some evidence. Well, it needs low pH to work, apparently, because it has to go through an
At least
endosome.
to start to work, but it
Yeah.
doesn't need it to work.
Right, but you do need to go through an endosome of the
do.
trypanosome. So the idea is that the trypanosomes can take up things by endocytosis, right?
Right through the flagellar pocket.
Is that where it happens?
It is where that
I think we
happens.
talked about this
We did
last
because
week.
we were attacking that with something else last time.
I've
with a-
totally forgotten. What
with
was that
a,
all
with
about?
a monoclonal antibody.
Oh, the monoclonal, right.
The cannelid.
By the
Right.
way,
Cannelid
Yes.
nanobodies.
Now Vincent has a mea culpa to announce. I know he does.
Yeah, I wanted to fix this. I didn't put it in the show.
Please do.
So we're talking about single-chain antibodies, but they're single-chain only in the sense that they just have a heavy chain, but there are two of them. It looks
Yes.
like a Y. They're two
Right.
heavy chains. That's how you make the combining site. But you don't need the light chain to make a high-affinity antibody, interestingly. That's the cool thing. So they're much easier to engineer
Yeah.
than having two different proteins, right?
And camelids have this already.
They do,
That's how we discovered it, you
which
said.
is because camels are mammals, right?
This is true, but they're very unusual mammals.
They are unusual mammals.
Dr. Seuss, right?
A camel is a mammal.
Or was that someone else?
No, that's probably Ogden Nash.
Is a camel a mammal?
Is it
And other
camel?
stories. Maybe
Right.
it's, I don't remember.
Right.
We did.
Oh, we're going to get a lot of email on this one.
It's fine. We love email.
We do.
Anyway, the first figure in this paper is a very cute figure which illustrates that ApoL1 will not lyse trypanosomes that produce SRA.
That's right. That's right.
But if you introduce the two changes into ApoL1, which are found in African American
G1 and
descent,
G2.
now they
That's
are unable
it
to be
reveals their
resistant.
cloaking
It kills them.
device.
It kills them, right?
Takes their cloaking device
lyses
away.
them.
That's right.
How would you measure lysis, Dickson?
Just watch them under the microscope.
Yeah.
But what they do here is they use a dye,
Yeah.
and it's a dye exclusion test, just like you would.
You
for
read the paper?
Stop it.
He even read the methods.
Stop
Must have
it.
read
Oh
the
my
methods.
gosh, I am
went into-
stunned.
I know these people very well because
Truly.
Truly.
I'm stunned.
Are you actually going to keep this on the air because I'm not getting insulted?
You
I really am getting insulted.
Oh,
I'm
it's-
surprised
it's,
you
okay,
read-
it's okay, Dickson.
You were questioning my veracity.
No, I wasn't. I just questioned that you read the
You're
paper.
questioning my velocity.
Your velocity. Okay. I keep everything in.
Of course, I read this paper.
Okay.
Of course, I'm deeply interested in these subjects. So I was even more curious than you thought I was because I want to know how you would do this kind of an experiment because you can't actually measure a membrane conductance difference in a single trypanosome. You can't do that.
This is
I
just
know,
the
I know, I
dye-exclusion.
know, I know, I know,
Alamar
I know.
blue fluorescence. Alamar.
It's a little
Yes.
bit different than the trypan blue
Similar
exclusion,
idea,
but it's
right?
the same
Got it.
idea. If you're alive, you can't take it up, and if you're dead, it goes right inside.
Okay. So the crux is
Yes.
they wanted to know if this protein formed an ion channel.
They wanted to know that. And why did they want to know that? Because they suspected that that's what's happening.
There was some evidence, I guess. right?
There was indirect evidence.
That if you poke a hole in a trypanosome,
they're gonna lyse it.
that
So
could
how
be
would you
done.
do that?
You could do that with an ion channel.
Because of the
right?
variant antigen on the surface, you can't poke a hole in any place except in the flagellar pocket. There's no other exposed region.
So if you made let's think of this a little bit. If you made a channel in a membrane, which is basically a protein that can make a pore, and we have
Yes.
channels that
Pore
make us-
forming.
We have all kinds of
proteins
channels,
exist all
right,
the time.
Daniel?
Yeah, we do.
We do. Calcium, potassium, sodium.
And some of them are in our heart, and they're very important, right?
True.
They're all important.
In our
They're all
muscle.
important.
So you make a channel in a membrane to allow
Right.
things. Some of them are controlled, and some of them
Yes.
just allow things to pass in and out.
Yes.
See, in the muscle, they don't have those channels. They have sarcoplasmic reticulum, and it's a multiplicity of like a labyrinth.
And so if you put a channel in a trypanosome where it's not supposed to be, you can imagine that stuff might leak in and out, and it could end
Can
up.
imagine.
The trypanosome could swell and
That's
pop,
right.
right?
And they do.
So if you have a isotonic environment inside the trypanosome, which means physiologically saline, right? And then you put a channel, what's going to happen?
Nothing.
The water will go in to try and dilute.
And the salt will come out.
The fly will get bigger and bigger and explode.
Yeah, but you said isotonic.
Yeah.
No, isotonic, nothing will
Nothing,
happen.
because outside of the fly is...
Hypertonic, that's what you want.
Hyper?
Or hypotonic.
Anyway, I don't know how putting a channel in
If it's
would do
isotonic,
it,
that's why we use 0.85% saline to begin with, because nothing lyses under those conditions, right? At least in our blood. And
Well,
trypanosomes live in our blood, so they are isotonic to our blood.
Yeah, nothing should happen, right?
Nothing should happen. So if you make it hypotonic or hypertonic to the trypanosomes...
I think they do speculate
They do.
later,
They
so
do.
we'll
They
get to
do.
that.
They
But anyway,
do.
how would you find out if something's an ion channel, Dickson?
I'm not a membranologist, so I think you would create an artificial membrane. I know what they did, so it's sort of preempting the discussion of the materials and methods. You can't do work on single trypanosomes, so you have to create a condition that mimics their lipid bilayer, and then it has a trans side and a cis side.
Yeah, so these are synthetic bilayers.
They're synthetic bilayers.
They're lipid bilayers.
Yeah, you can buy the lipids and make them, and then you can
put your protein in it, right?
You can see if the protein affects the lipid
You
bilayer.
add it to it, right?
You use these little electrodes. One is on each side, and it's connected to the membrane by agarose. It's a very clever way of doing all these experiments.
Is this a patch clamp?
It's a little bit like a patch clamp.
Basically, it's a very tiny...
It's an enormously small patch
and
clamp.
you can measure the conductance
and
across.
You can measure... you can do electrophysiology on this.
So if there's no holes in the membrane, there shouldn't be any conductance, or very low.
That is exactly
and if you put
right.
pores, then you're going to get conductance.
Right, and what they found was...
How did they set it up? They had different pHs on either side,
They did.
right?
They started out at low pH because they imagined something getting inside of a trypanosome would first of all have to be taken into the endocytic vesicle, and that's usually an acidified vesicle
Right.
that comes about as a
So
result.
on one side they had low pH, and the other
Right.
side neutral, right?
Right, and then they
They got a
raised
little
the
bit. They
pH.
got a little bit of conductance,
They
right?
did.
But they did another thing that made them get a lot more.
Oh yeah, they just changed the pH to a basic, and whoop.
So they add the protein at low pH,
Yeah.
and then they neutralize the pH, and boom,
right.
goes
But
way up.
why are you directing your attention to me? He's the biochemist.
Are
I'm
you the
sitting next to a
I'm sorry.
biochemist here. He's
Sorry.
bursting
Apparently,
with
no.
knowledge.
Are we okay so far with this?
Yes, and I thought it was great. I often start with materials and methods, and how do they go about.
That was interesting.
And I thought it was great the way they create an artificial membrane.
Yes.
And what they do is they discover, they show to us that at a low pH, this ApoL1 inserts into this membrane that they create. But then when they alkalinize it, then it really opens up and the activity increases thousands-fold.
Sure.
And they have a great figure that, why this is in an endosome. So why does that even matter? Does that just mean more stuff gets in the endosome? But when you think, and now if you're driving, keep paying attention to the road, to our listeners, but try to imagine this. What are endosomes? Endosomes are a portion of the membrane, that's the outside of the cell, and it then invaginates, creates a little ball, right? Now, at this acidic pH, this ApoL1 is going to insert into the membrane. Mm-hmm. But then what's going to happen is that little ball is then going to come back and refuse with the membrane. Right. So now you have a channel that's going to allow all this water to flow in from the outside. But not only do we have a certain amount of sodium inside these trypanosomes, but there's a lot of osmotically active agents, a lot of proteins. So when this happens and they recreate this in the experiment, you actually see these trypanosomes start to undergo this swelling phenomenon, and ultimately resulting in their death.
So their idea is the channel forms in the endosome, low pH is required, then it alkalinizes, it goes to the surface, it's recycled, the channel is now in the plasma membrane, and there the pH is neutral, and now it's opening up, right?
Exactly.
You got a hole
and you're
when
getting
you didn't
all
have
the
a hole
water
before.
pouring in.
That's right.
There's a hole in my bucket.
Mm-hmm.
You know that old nursery rhyme?
There's a hole in my... There's a song by that name, too, by the way.
So they did a cool experiment to give this some physiological relevance. It involves SRA, Do you know?
Yeah, no, they started to
You know
add
what
the
that
right
is,
reagents.
Daniel?
to
See,
it.
I'm looking at Daniel.
Now I get it.
Thank you.
Probably mad at me now.
But I actually knew what you were going to say.
You want to tell us about that, Daniel?
But no.
No.
No, he's
I'm a big picture guy.
he
So,
just went back to his MD mode.
If this is really relevant, this low pH assembly, neutral
Yeah,
pH,
that's right.
what would you expect SRA to do to it?
Aha.
If you added SRA to the...
Well, what the SRA does, and they show this, is that there's a certain portion of this protein that is inserting into the membrane. And that was actually that was what gave them this idea that this had homology to other things that insert.
Probably a lipophilic end.
And this lipophilic end can be bound by the SRA. And when it gets bound by the SRA, you're going to no longer be able to insert it into the membrane.
Right.
But by slightly varying the sequence where the SRA binds, that's our G1 and G2 variants, the SRA can't bind anymore.
Exactly.
And so they show that.
SRA blocks conductance. Actually,
Yes.
it allows formation of the low conductance, but then it doesn't open.
Right. You get no pore.
And then putting in this G1 and G2 variants, which are from people, that negates the effect of SRA. All
Yes.
in this lipid planar
You got
bilayer
it.
system. So
yeah,
that
an artificial
it
membrane
makes
system.
it relevant, right?
It does.
brings it
Yes,
back
it does.
to the biology.
Well, you
which
can
I think is
explain
really
all of the things that occur in vivo, but you're still stuck with knowing what the pore actually looks like on the surface of the trypanosome.
how would you do that? That's because these
Maybe
are tiny,
you could do
right?
that by a cryo-electron microscopy. If you had markers for the membrane proteins, you could see whether or not they were parted and whether the same molecules were still present, right? You might even be able to watch it happen if you used high-voltage electron microscopy.
I think that would be hard because these are very tiny.
You know, at 1,000,000 volts, you can actually watch living cells behave under the electron microscope.
Should I watch you behave at 1,000,000 volts?
Not at that level. I would be deader than a doornail with 1,000,000 volts passing through me. I think you would not see me.
So the morphological changes that happen, which precede trypanolysis when you add ApoL1,
Yeah.
a rounding of the cell body,
There you go.
swelling of the cytoplasm,
There you go.
and then complete lysis. So it is their opinion that cytoplasmic swelling is a result of osmolyte influx.
There you go.
It's a defining feature of a trypanosome lysis.
So that means then, if that's correct, and I'm sure it is, that the trypanosome is constantly fighting this by extruding things like water that keeps fluxing in. It has to keep getting rid of it. So it has a very active transport system.
But it fights it by SRA. It just blocks the hole.
No, I don't mean that. I mean just living inside human serum, for instance.
You know what? If you're going to live in someone else's bloodstream,
You better be able to do that.
Ready.
You better be ready.
'Cause-
So imagine where this parasite lived before that, though.
Where did it live before?
Well, it lived in the gut tract of a tsetse fly, for instance, or in the salivary glands of a tsetse fly.
Probably-
So
probably
those are
had to do
quite
things there
different
as well.
conditions, aren't they?
It's probably doing other things.
It's
that we don't
highly
know of.
adaptable. It's highly adaptable to these situations.
You always wonder how these evolved. You know,
Yeah,
how did
it's-
it evolve to be able to do what it needed to do to
Exactly,
exist in the fly,
exactly.
but then at the same time evolve to exist in its mammalian host?
So some of the trypanosomes, well, they're not actually trypanosomes. They are precursors to the trypanosomes. Actually, are infections of plants.
Really?
Yeah, the milkweed plant has one of
Interesting.
those. Yeah, so they always used to look at that as a throwback to the ancient days before there were mammals. That plants had parasites that insects could transmit between them by the same mechanisms that you're looking at now for the adults of.
And maybe the mammals acquired them by eating plants.
There were flies by that time, so there could have been vectors. That,
Daniel, how could you imagine this channel having anything to do with kidney disease?
You know, they
What's
actually,
happening there? What's
they
happening there?
they try to speculate, and you know, with the idea that their advances, their discoveries, feed into that. And if we think about this protein, what is it doing? It's inserting into cell membranes.
Yep.
And how selective is it? Is it going to also insert into our cell membranes? It's going to insert into the cell membranes of the kidney cells and potentially cause them to undergo. That's what they're speculating.
So I have a question, and relating to this, the normal role for apolipoprotein 1 is to transport cholesterol. Is that correct?
In the blood, right?
So in the blood, but it's taking it somewhere, isn't it? So what happens when it encounters a cell? What is the receptor for that combined molecule?
Well,
and then what happens?
So there is a receptor for HDL, right?
Yeah, there's actually each one. There's the LDL,
and
receptor,
HDL.
the HDL receptor.
And
So
did
what would
you know,
happen-
Daniel, that the LDL receptor is also a receptor for rhinoviruses?
Get
Yes,
out.
yes.
Get out. How
You want me to get out? This is the
ingenious.
second
No,
time.
this is an
You've told me
expression.
to
I'm
get
just
out.
using. I'm trying to bring this up.
Three is a... what is it called?
Three's a charm.
I'll leave. Yeah, so then it's taken up by endocytosis, and then the lipid is separated from the carrier, right? And then it's recycled back out again, right?
Yes.
And is that what happens with cholesterol?
How interesting. This
Yeah.
is just going through the same cycle in a fly or a human, but it's making this channel.
about
Sorry,
that?
in the tsetse fly, in the
Oh,
trypanosome, sorry.
It's trypanosome.
It's doing the
That's
same
right.
thing that it's
That's
normally
right.
doing, but now it's making a channel.
So
So I
the
wonder in
apolipoprotein-
people if it makes channels when it goes through this, right?
Well, that's the concern in the glomeruli. I don't think normally in the liver uptake, but this is great. You know, one of our writer inners, one of our listeners, one of our emailers, talked about the fact that we don't probably discuss enough biochemistry. But the interesting thing, you bring up the LDL receptor in viruses, but our innate immune system, like a lot of other animals' innate immune systems, the B1, the innate cells, about 10% of them target what? Oxidized LDL. Why? There's a certain homology. If you can bind oxidized LDL, you can also bind moieties that are common on a lot of bacteria. So a lot of this whole lipid metabolism has a tie-in with our immune response.
The normal uptake of HDL and LDL, it shouldn't be forming a channel.
No,
right?
no, and I
So
don't think it is.
This is specific to the trypanosome.
I think that's true. I think the APOL1 is actually unique, and I think unique to humans, gorillas, green monkeys, as we mentioned. And I think it actually evolved not just because it's necessary for the lipid transport, but I think what nature often does, what evolution often does, is take advantage of something that exists, modify it in such a way to have some secondary use.
It's interesting. And maybe these variants have some channel forming activity in the kidney, and that's why it's a bit of a problem.
Yeah. I mean, that's where it's produced, so there may be some issue there, and it also makes you speculate if certain levels, for instance, if the heterozygotes do well, is there a therapeutic potential for this? Someone who's sick with these trypanosomes, can you infuse certain levels of G1, G2 variants that can't be neutralized by the trypanosomes? Can these potentially be, medicines that we could use?
APOL1 is a minor apoprotein component of HDL, which is made in the liver. It's found in the vascular endothelium, liver, heart, lung, placenta, podocytes, proximal tubules, and arterial cells. So it is in the kidney. Protein has a secreted form that allows it to circulate in the blood, forms a complex with high-density lipoprotein particles that also contain apolipoprotein A1. It is a member, blah, 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 blah. It doesn't say anything about channels, Trypanosomes endocytose, the secreted form of APOL1, forms pores on the lysosomal membranes of the trypanosomes, which cause an influx of chloride. So that's actually a little bit wrong. It's not in the lysosome. It's actually on the surface of the trypanosome that the pores
Yeah, that's
are
how it
formed.
actually acts. And I thought that was nice in this paper. They have a figure 5 where they show that, okay, even though the insertion is occurring in the endosome, the functional lysis is only once that endosome then refuses with the surface
So
of the trypanosome.
these channels that they formed, I mean, the lipid bilayers that they make, these are made, I'm going to tell you how they're made. They make bilayers from soybean lecithin,
Exactly.
which you can buy from Sigma,
Right.
and they remove the nonpolar lipids,
Right.
and they just form this across a 80 to 120 micron hole in a Teflon partition separating two aqueous solutions.
Yeah.
So this protein will make a channel in that. That is a completely generic membrane.
exactly right.
So why doesn't this protein make channels in human cells? Maybe because it's secreted, but it is taken up by endocytosis, right? So why doesn't it make channels? They don't address this in the paper at all.
Is the
No,
protein
I-
actually destroyed when cholesterol is taken up? when the complex is taken up by a cell and the cholesterol is released from its carrier molecule, apolipoprotein 1, is the apolipoprotein 1 destroyed?
I would guess it's recycled back out to the surface, it goes on. But
I bet you there's somebody out there. That last person who wrote in for the biochemistry,
he's going to tell you that you should know.
I bet they no, because that's not my field.
HDL.
Ever was, never will be.
Let's look up HDL, Dickson.
Okay.
High density lipoprotein, which, by the way, is a beautiful looking molecule. They actually have and here's a little figure of what happens to it.
Okay, then.
High density lipoprotein.
Because you continually make it. I know that. And
see what it does here. Structure and function. Blah, blah, blah.
Yeah, while you look it up, they actually they talk about some earlier work that the ApoL1 can actually trigger autophagy in human cells. And so, and they're, I think, suggesting that,
and then apoptosis.
We know what triggers that.
Okay. Interesting. Is that why it's not so good to have elevated levels?
Well, that's the concern. People who are homozygotes for these G1 and G2 traits.
Hypercholesterolemia and stuff like this.
HDL transports cholesterol mainly to the liver.
Okay.
And other organs, steroidogenic organs.
Yes.
It's removed by receptors, which mediate the selective uptake of cholesterol from HDL. Okay. And there are several pathways that result in that. And so the HDL could come out and be recycled, and eventually it will degrade. It has a half-life, right?
Sure.
Mm-hmm. But there's no need to destroy it to get the cholesterol off. That's
I just thought it was endoscytosed, and then it was, somehow gotten rid of. That's all.
I don't think it has to be gotten rid of necessarily. Okay. Anyway, that's we are pushing the limits of our knowledge.
We are.
Obviously, my biochemistry is
it was
horrible.
an open question. That's all
I just want to know why it doesn't make channels in us. Maybe it does. Why would it specifically make a channel in a trypanosome if it can do it in a lipid bilayer that's pretty generic that we've just discussed?
But our cells are not generic lipid bilayers by any stretch of the imagination.
Maybe it's inhibited for some reason.
Yeah. Well, they actually referred to a paper in 2008. I think it was reference 42, and where they talk about the fact that maybe it doesn't end up getting recycled back to our membrane, but it may, this autophagy I mentioned, it may, because it inserts into our endosomes, start to trigger an endosomal-based apoptosis.
Well, the people with people with G1 and G2 mutations do develop kidney disease, and that may be the answer to your question.
Yeah, well, it may be making channels
that you can't repair.
So
and I think ultimately we don't know yet, but
right.
It is an active area of research, and they're suggesting, hey, this might be a hint.
How many people have these two mutations? Do you know? There was a paper that just looked at this, which I've closed, but
there was the paper in the New England Journal. It was, I want to say, December of 2013, and that probably would have a frequency of these.
So here is a paper in PNAS by some of the same authors.
Okay.
Evolution of the primate trypanolytic factor ApoL1, where they actually looked at several thousand people, I want to say. We surveyed 1,700 Africans for the presence of G1 and G2. And what did they find? In different parts of Africa, it varies, but it's also a small. So they have a map of Africa with pie charts, right?
Oh, great. Wow.
And G1 in some areas is 50% of the sampling. Wow. In Western Africa, but in the East, it's mostly wild type. There's a little smattering of G2 and G1, very small. Does
that mean that most of the population in West Africa is resistant to trypanosomiasis?
Well,
if you say 50% has a certain resistance.
That's a huge selection.
Yeah.
I think that goes along with what we were speculating, that since the Western African trypanosomiasis is much more common, affecting the human population, we would have expected much more of a selective pressure. So that makes sense.
Amazing.
I'd like to know if the Neanderthals had this mutation.
The Neanderthals didn't live in Africa.
I would say
they
no.
They didn't live there. They lived in Europe. They lived during the Ice Age, so they didn't encounter this at all.
Well, I think also
not even a little bit.
They tried, they go back and they try
That's
to
why
guess
I want to
it.
know.
Well, you just found out.
No, they try to speculate, and they have certain ways of doing this, of when a gene mutation gets introduced
you
into
said
the
10,000
population.
years
And they're
ago.
suggesting 10,000 years ago.
Yeah, that's based on mitochondrial clocking. Is that right?
You see, the most interesting part of a paper comes after you've finished it and you start thinking about the implications.
Right.
And that is what
Because you're looking at this like maybe it's a potential cure for the disease. If you could
How
engineer
so,
something
Dickson?
that looked like
how would you do?
well,
use it?
if you were clever, maybe, and
All right, that rules me out.
That rules everybody out. Do
you
the
shouldn't
people
speak for Daniel?
do people with the G1 or G2 mutation transport cholesterol the same?
Mm-hmm.
I just want to know that first, because if they don't, then what I'm about to say doesn't apply. Because
But you know what, Dickson? You could design a channel blocker that would hit the trypanosomes.
well, why
that would
haven't they
protect
done
them.
that yet?
That would protect them. Maybe you could give
right.
people this channel
Right,
blocker.
right.
If in fact
Yeah, you could counter
the
the effects on their
channel
kidneys.
is causing kidney disease, then
Right.
you could make a channel blocker to
That's-
prevent that.
that's
right?
Or maybe a dose toxicity. If you could give people this ApoL1,
Sure.
constructed variant at a certain dose.
Yeah, that would
And maybe
work.
even just during infection. I mean, someone has infection.
Even during infection, that would be great.
Let's do gene therapy.
If you did this during
Adeno-associated virus
infection,
delivery.
yeah, you just give them an injection of modified apolipoprotein 1, which overcomes the SRA from the trypanosome, and you cure them. Because these are not in the tissues; they're all in the blood.
You have to give it in a vector because giving people a protein is difficult.
Well, if you're dying from trypanosomiasis, I think I'll be the victim.
right.
Give me some of that.
Okay, what's next, Daniel?
Well, I think we have another case.
Okay.
We do.
Are we ready for that? Is everyone ready? I'm ready. For
On
Now
the
listen,
edge of my
Dickson,
seat.
take it easy with the crayfish this time because you gave it away.
I'm not going to say a word.
Now, this is a case. We start off, and people are probably familiar with the format by now, hopefully, or we'll have newcomers. Welcome, newcomers. History of present illness. This is a 36-year-old woman who came into the office. This is when I'm private practice in Colorado. We're moving cases out there. It's August, and her chief complaint is diarrhea that's been going on for 5 days. She reports that the stools are watery. She's been having about 4 or more per day. She notices that there's some mucus in the stool. She reports stomach cramps, nausea. She feels like she's got a low-grade fever, and she says when she checks it at home, it's a little bit of 100. And she reports that no one else is sick.
Daniel, this mucus in the stool, how the hell would you know? It's such a mess when you have diarrhea.
Well, not if it's watery, it's not. Then you can notice what they
What
would
do you
call
know?
a mucus cast.
What does that mean?
It's strands of mucus that pass out, and they resemble things that you think they are, but they're not. Like
I guess
worms,
I've been
for
fortunate
instance.
never to have mucus in my
Well,
diarrhea.
that's right.
I've never.
That's exactly
I
right.
can't.
So
So it's
what
really
do you mean
ropey looking.
It is interesting
looking
for
stuff,
our
yeah.
parasitic aficionados. Sometimes it looks like a tapeworm, right? They look
Exactly.
down, and sometimes it can be
Okay.
confused for it. Sometimes people bring them in.
Oh.
And look what I found in my
I've seen a
stool.
lot.
And you say, yes, that's mucus.
I used to
but-
take a lot of you-know-what from people.
Yeah.
Okay.
No, and we try not to be arrogant because, we're supposed to be experts in looking at feces.
Right.
You're not supposed to, as a,
What do you mean no one else was sick? You mean no one else? I don't want to see the
Okay.
things. But
Yeah, I saw Dickson trying to peek at my...
No, I didn't. But when I was pointing to his case history and asking when she said no one else was sick, did she mean no one else in her family was sick, or she knew no one else in her immediate surroundings were sick, like at work? Or what was she referring to when she said no one else was sick?
Yes. She is, well, this gives us a little bit of the social history. She's married and has one child, a
Right.
4-year-old son.
Okay, fine.
Her husband's not sick, her 4-year-old's not sick. So when she says no one's sick, she's referring to people in the household
Or her pets?
or her pets. Does she have any pets?
Innocent question.
No, she doesn't have any pets.
No pets. Okay.
No pets.
Does she have a fever?
She
Said
tells
she had
us
100
she tells
degrees.
us that she did at home. She said when she measured at home it was a little bit of 100 degrees.
All right.
And when we do the physical, actually, I'll give you that ahead of time. She actually does have 100.1, so just a little bit. And that's always interesting. If you use Fahrenheit, that's a fever. If we use Celsius, then we wouldn't worry about it because we like big round numbers, but it's over that big round number. Blood pressure normal, heart rate's fine. While you're talking to her, you're looking at her. She looks okay. But yep, she gives you this history that we got.
And may I ask where she lives?
So this is Fort Collins, Colorado. She lives in Fort Collins, Colorado.
In Fort Collins, not on a ranch. For
No, she lives in Fort Collins, Colorado.
Okay, fine. She lives. She's a city dweller.
Sure.
I wouldn't call that a city, but they might.
It's one of the mega cities of Colorado.
Right.
Population, what, 95,000?
Yeah. All right.
Yes.
But at least she doesn't live on a ranch.
A lot of good microbrews.
There's more of the CDC,
CDC is there.
ID labs are out there
That's
in
right.
Fort
Vector.
Collins
I meant
than
beer.
really anywhere.
I meant beer.
Beer?
It's the home of the Vector Labs.
Yeah. Well,
Really?
So that too.
Yeah.
Those are the high points
that's where
too.
Dwayne Goobler
Is
ruled.
more beer is brewed in the region between Fort Collins and Golden, Colorado than anywhere in the world, more than the combined Germanys.
So I
Wow.
probably told you that. That's
No wonder there's a
remarkable.
drought.
We had a virology meeting there last summer, and the guy who ran it was Jeff Willis, who's a microbiologist at the university, and he had a local brewery make us a custom brew for
my
our meeting,
God.
which was distributed free of charge. He gave
Really?
us pint glasses, and it was
Okay.
called Hepatitis.
I like that. Do you know who the brewer was?
Yeah, I do, but I don't remember. I'll bring it in.
Okay. No, I had good friend there. Actually, the two beers I'll recommend for people going to Fort Collins is there's the Fat Tire, the New Belgian
That's
brewery.
good.
Yeah,
I like
Fat
that.
Which is a
Tire,
great
yes.
one.
I like that beer.
And a friend of ours, Katie, was the quality inspection person there. And then there's another, Odell's. Not O'Doul's,
I've
but
heard Odell's,
Odell's.
yeah.
Don't
And they make great beer there. And actually I should say one of my weddings, they made me a custom
One of your weddings.
beer.
Okay.
not too much personal TMI here.
But no, they make great beers in Fort Collins.
Interesting. I would never have thought of that as the beer capital of the world, but of course, Coors gets all of its water from the Rocky Mountain rivers, which I take exception
sorry-
to. by the
sorry
way.
for that
I'd rather
diversion.
fish it than drink it.
No, so let's get a little more. Do you guys
Yeah,
want to know?
more.
Do you want to know any more about this young lady?
I'll let Vincent do all the asking.
Okay.
Travel history?
She has stayed in Colorado. Doesn't report any travel outside of the state in recent time. Again, could she potentially get a parasite not leaving Colorado?
David Dickson, you had talking to the mic.
Of course you can.
222 is the brewery.
Okay. I don't know that brewery.
TWO22.
Oh, really? Okay.
Yeah.
I don't know that brewery.
Yeah.
So it's only she's only been sick for 5 days, right?
Yes, she says the diarrhea has only been going on for 5 days.
She ever had this before?
She's had diarrhea before,
Yeah.
but
And
for this
never.
duration?
Yeah.
It's never this long.
No, 5 days is a little longer than
is
I
long.
think she's had it before.
And, and she has not left Fort Collins for a few months.
Well, she's left Fort Collins, to do the normal things that people in Colorado do in the
She
summers.
hasn't lost Colorado. She hasn't
Herd
left
cattle.
Colorado.
She hasn't left Colorado.
And sorry, pets? Do we have pets?
No pets.
No pets.
Do we farm sheep or anything like that?
She's not on a farm. She
Does
lives
she
in
ride
Fort
horses?
She does not ride horses.
Okay. And so she's been sitting at home. Has she changed?
Oh, she hasn't been sitting at home.
She works.
She does work.
Where
bet you-
does she work?
She works in the home, which often involves more work than those of us who
Oh,
work
she's
outside the home.
Works in the home,
What does
okay.
she do in her home?
She has a 4-year-old son.
Okay, and her
Takes care of
4-year-old
him.
She's a
son
stay-at-home mom. They would
does her
say stay-at-home mom,
4-year-old
but
son
that's a big job.
go to daycare? Or is
He does. He does go to daycare.
Mm-hmm. And his son has been fine. Anyone
What
else?
does her husband do?
Hey,
Husband actually works for one of these tech companies,
okay.
and he's been fine.
Now, at the daycare, have there been any other outbreaks of diarrheal disease?
No, not recently. There periodically are.
Have they been camping?
They have been. They have been.
Oh, really?
No
gonna-
crayfish.
I'm going to shut
No
up. now.
crayfish.
They've been
I'm going
camping?
to shut up now.
What did they do?
No.
in this?
I think you can keep asking questions. This may be the time that Dickson gets it wrong. But you are. There's been no raw crayfish
When was
ingestion.
the last time she camped in relationship to when she got sick?
So she tells us that she had a recent. This is like we said with the Missouri, what people do in the summers in Colorado. A lot of people go backpacking.
They do.
And she said that she enjoyed a trip to southwestern Colorado, the Durango area.
Durango.
Exactly.
Yeah, I like it there. Nice.
That's
It is-
fabulous.
it's
a
beautiful
fabulous
there.
area.
Good
Yep.
skiing in the winter.
Great trout fishing in the summer.
And she was there about 3 weeks before.
3 weeks.
And she was there with her husband, her son.
3 weeks.
And she describes the trip to you.
Did she drink any water from the rivers?
They did, but they treated everything with iodine.
Iodine. Just iodine? They didn't boil it, or they didn't filter it through a millipore filter?
Not the drinking water.
Not the drinking water. Just iodine tablets.
And they saw lots of animals, right? Because that's always the great thing about when you're, it's great we're worried what's wrong with her, but it's always nice to, how was the trip?
Right.
And she saw a lot of animals. They saw a lot of beavers, deer, elk. They're a little upset that there were like too many darn cows, and there is an issue.
It's true.
We discussed it a little. I remember
free-range cattle.
Well, there's a lot of land out there, Bureau of Land Management land,
yes, that's BLM.
That the ranchers pay a certain fee, and they're allowed to have their cows in that land. And actually, that's part of, I think, the establishment, right? When they establish these lands out there for timber, for ranching.
Yeah.
And it is, and she complains, and we, about the fact that there's too many cows. You don't really want to see cows when you go back.
That's right. It takes away from the pristine illusion of pristineness.
Exactly.
I'll just say illusion of,
and it's all over the place. At one point, her son, wants to go at this beautiful stream, and next thing you know, he slips and he's down covered in cow dung.
Are you telling us that? Well, that's let's see that we've asked too many questions now. I think
that's okay. And I mentioned the beavers. And that's one of the nice things about the Southwest is that a lot of these animals are coming back. So as you hike, you see these beaver dams and the lodges. And
yes.
I would like to make this a little harder for our listeners.
It might be already, and you don't know it.
No, you might think you know the answer, and you might.
No, I don't know the answer, but I would like our fans to.
The checklist of possibilities goes up as the number of possibilities outdoors goes up.
Of course.
So we've already got a big list.
So she had contact with cow feces, but any other contact with any of the other animals?
Well, as we mentioned, she's drinking water out of the streams. There's
and there are beavers
there.
In these water.
Okay. And they warn you not to do that, by the way. So that was like in direct violation of a warning, unless she thought she was treating it with iodine tablets, which.
I went on a long canoe trip upstate a few years ago, and we had to treat our water because we couldn't carry enough. Right. We had quite a few.
But you had lakes to go through, right? I mean,
so we had a filter, and we would put it through the filter, and it would go in brown or green. It would come out crystal clear.
Right.
But you could still taste the different waters depending on where you were.
That's right. Yosemite Park, my wife and I did the High Peaks hike one year, and we did the same thing. And you'd throw the end of the device into these yucky-looking pools, and you'd start pumping like crazy. Not much comes out, by the way, so you really have to pump a long time. But eventually you got enough to drink, and it did have a je ne sais quoi about it.
But it was a very fine filter. So
so I must warn everybody that iodine tablets don't eliminate a lot of things. It will eliminate Shigella and Salmonella and things like this.
Yeah, I would filter it.
It doesn't touch the organisms that we've been talking about. It doesn't touch them.
Well, I'm going to give you guys a few more laps because I don't want you to think you know the answer and then next week say, "Ooh."
Oh, I don't know the answer at all.
No, I never pretended to know the answer.
Dickson clearly does, but
no, he does not. Clearly.
So we ordered fecal, blood, and leukocytes.
Right.
Those were both negative.
Sure.
So no white cells, no blood.
No.
The CBC was normal, so that's a complete blood count. So we're not seeing an elevation in the white cells. We're not seeing anemia. We're not seeing changes in platelets. The chemistries, electrolytes, kidney functions were all normal.
No fats in the stool?
Stool fats was not ordered. But I'm going to give one last thing that I think will be helpful.
Uh-oh.
I'm going to say 3 stool ONPs were ordered, and they were all negative.
ONP?
Yes.
Ova and parasites.
Ova and
they were all negative.
Okay.
So that's what I will leave our listeners with.
Right.
Good. I think that's a good place to leave them.
I do too.
You're just dangerous, Dickson.
I'm bursting at the seams with
when we
questions.
finish, we have a few emails. Would you like to read a few?
Yeah, sure.
Dickson, do you have your...
I actually do.
And Daniel, you both can read emails.
We both can. And so here's one by Andrew.
Take it.
Andrew writes, "Hello, TWiP. My name is Andrew Vahi. I'm a 23-year-old programmer who works for a tiny company named Menards. Actually, we're third largest home improvement company in the U.S., but that's not important. I was writing today to mention that I listen to TWiP and TWiV as often as I can. I greatly appreciate you three take your time to supply everyone with so much in-depth information. I'm also a huge fan of the new reboot to the TWiP show. Daniel is a great part of the team and really meshes with you two. The case of the week is by far my favorite thing at the moment. I know nothing about the medical aspects, but I thought I knew this week's answer. Dracunculiasis, guinea worm disease, was/is my guess. This was until Daniel started to explain that the gentleman wasn't feeling any pain. I look forward to hearing more information about this case and many more in the future. But, it's currently minus 11 degrees Celsius, and when I get back to work today, it was minus 22 degrees Celsius.
All right. Well, he's already heard, presumably, what we had to say about that.
I think so.
Yes. Let me read the next one. I like the next one.
Please do.
So Mike writes, Dear Doctors Racaniello, Despommier, and Griffin. Did I pronounce this correctly, guys? All right.
Despommier. That's close. You were close enough. You were close enough.
First, let me express my great appreciation for the effort you put into these excellent podcasts. I discovered TWiP, TWIM, and TWiV a month ago, and they quickly became my daily routine while driving to lab. With so many episodes, I'm like a kid with a giant ice cream sundae too large for me to eat. Fortunately, you seem less likely to melt. As I am an immunology graduate student focusing on the immune response to H. capsulatum, I feel the only thing lacking is a TWiF podcast this week in fungi.
Right.
Yeah, I've had similar thoughts, Mike, actually.
Do you know anything about fungi?
I'm supposed to.
Hardly anybody knows anything about fungi. They're
love to do it.
rare birds.
Fungi are challenging.
They are rare birds.
And I think that people could use a TWiF.
Well, there's a guy at Einstein who would be perfect, but
Yeah?
he's too busy.
It's too busy.
Arturo
How can you be too busy for this?
You could be too busy for this. I would like to make a guess for the recent TWiP case study, filariasis caused by Wuchereria bancrofti worms clogging up the patient's lymphatics. The dramatic, chronic, non-pinning, painless swelling of the lower leg is very typical. The open lesion is not so typical, but could be caused by a secondary infection. Blood smear would be diagnostic and cheap. Connecting this guest to the earlier TWiP podcast describing M. bovis and Cape Buffalo, I would recommend the recent paper, Filarial infection modulates the immune response to Mycobacterium tuberculosis through expansion of CD4 positive IL4 memory T cells by Chatterjee et al. And this is
Journal
in
of Infectious Diseases.
Journal of
Right?
Immunology.
I think
Journal-
it's Journal of Immunology.
Journal of Immunology.
And I think I actually remember looking at that paper. So we can discuss whether or not. But patients with filariasis had an increased percentage of CD4 T cells producing the cytokine IL4 in response to MTB antigen. IL4 is a Th2 cytokine and maladaptive in tuberculosis. And this is when he gets to the thick of it, I guess, here. Finally, I'd like to recommend that your podcast delve a bit more into the shifting labyrinth of CD4 T-cell polarization. The generalization that Th1 cells induce cell-mediated immunity, while Th2 cells are immune regulatory and or induce antibody-mediated immunity, is somewhat outdated. Yes, I agree with you. Th1 site. We got to keep this simple. No, we don't have to keep this simple, and I am going to agree with you here. Th1 cytokines are important for controlling intracellular pathogens and promoting IgG2A. Th2 cytokines are important for clearing helminths and promoting IgE. However, the role of Tregs for immune regulation and T follicular helper, TFH, for antibody production are needed for the full picture. Perhaps you could convince an expert labyrinth navigator to join you for an episode. Actually, and you mentioned John O'Shea would be great. And John O'Shea has a great, he did a review a few years back that I used to keep like on my desk. And he breaks down all the different subsets, Th1, Th2, Th17, the T follicular helper, et cetera, and it is becoming et cetera. So thank you, Mike. That's a great email from you.
Mike is an MSTP student.
How about that?
In Cincinnati.
How
Yeah.
about that?
Our friend Varun from India writes, greetings, professors. Following up with the case TWiP 83 looks similar to elephantiasis. I would ask for blood counts and absolute eosinophil Giemsa or Leishman stained blood smear for sheathed microfilaria. Would also look for a wet blood smear to look for any slashing motile microfilaria. Wish I could see the patient's leg photo. Wuchereria. How do you say it? Wuchereria.
You're going to get me to mispronounce it eventually because I'm not used to hearing it.
Wuchereria Bancrofty. Thanks for all the shows. I simply love TWiP Reboot. You guys are simply awesome. Dickson.
Dear. Okay. Adam writes, TWiP team. Is elephantiasis too simple of a diagnosis for the case presented in TWiP 83? The fact that the scrotum did not swell makes me skeptical, but maybe the worms didn't impede lymphatic flow enough proximal to that area to cause swelling, which raises an interesting question. Is gravity, in opposition to lymphatic flow, ultimately the reason why elephantiasis usually occurs in the lower limbs? I think the worm itself is negatively geotropic, and that's why they locate in the lower limbs, but they don't always do that either. My mom was a clinical microbiologist at UC Irvine, and I will never forget flipping through one of her books when I was no more than 10 years old. I came across an image of a man with a gigantic leg and was transfixed. Later, she had to explain that this was what was called elephantiasis. With my youthful imagination, I promptly decided that this condition was caused by a disease spread by elephants. I suppose knowing the actual science behind it, parasitic life cycle physiology, etc., is equally fascinating. Short aside, I teach middle school science in Chicago, and your podcast, Trifecta, keeps me intellectually stimulated, makes my commutes go by quickly, and gives me lots of cool things to share with the kids every single week. It's snowing here now, but you probably could have guessed that.
Nice.
Cheers, Adam. Well, I have an addendum to add to Adam's humorous remark as a 10-year-old, and that is he lives in Chicago. Gary Larson is from Chicago, and Gary Larson used to write very intellectual jokes that only certain people could get. And so he showed an elephant cocktail party at which an elephant showed up with one of its four legs very thin, and one elephant that was not part of that syndrome was talking about him behind his back, and he says, you know, he's got humanoniasis.
That is great. That is great.
That's very good.
Yeah, it was great.
Humaniasis.
I saved that joke. I put that in our lab.
You have to send me that cartoon. That would be great.
I'll find it. That's easy to find. Easy to find. He's got a bunch of them for parasites. He was really a very sharp guy, and he still is.
There's one where there's a party. There are a bunch of microbes at a party, and one of them is doing something, and everyone's pointing and saying, "Look, Vinny's mutating." So I used to keep that on my wall.
Cool. Very cool.
Daniel. We're up to Suzanne.
Suzanne writes, I'm sure someone else has written in, but I believe the character Klinger was supposed to be Lebanese. I do remember he was from Toledo. I remember that too. And a fan of the Mud Hens.
I knew.
He was one of my favorites as a kid. Actually, I might have counted the whole cast as favorites. I love the show and learned a lot from it, from what kimchi is to being more tolerant of others, along with a good respect for the science of medicine and surgery. It was a great show to grow up with.
Yeah, totally agree with that.
And that was a short one. Let me read another. Tim writes, first, thank you for the Twix podcasts. I enjoy them all. Second, it is great to see the TWiP crew expanded with the addition of Daniel. I also very much like the new Case of the Week segment. Alas, this week's mystery parasite has me stymied. So we have one person that we stymied.
Okay.
But I find it intriguing as much for learning more about the diagnostic art as for the specific parasite. Third, yes, in a past TWiP, Dickson did explain at length why the P in TWiP should be parasitism instead of parasitology.
Oh, my.
It seemed to make good sense at the time, although I cannot recall the specific episode.
Tim,
does it still make sense to you?
Dickson.
Okay, fourth.
It's okay. It's okay. Mea culpa, mea culpa.
You always argue with me.
I said mea culpa.
No.
That means I'm sorry.
It doesn't matter.
I have to say it in Latin, though, because I can't actually come out and say I'm sorry. But I'm,
okay, fourth, in this last TWiP, number 83, as has happened at one time or another on all the Twix podcasts, there were once again some questions raised which took the form of why questions. Oh, yeah, I remember reading this. And in response, there was the admonition that, to paraphrase, we don't ask why questions.
Right.
I find this apparent proscription of why questions across the Twix realm, and presumably therefore in biology in general, troubling.
I would agree with that. I hope I was never the person that said
You
why.
always say we can't ask why questions.
I do say that. That's correct.
There's a reason, though, in biology, because you don't know why a thing is. You just know about it and how it functions. But why it is, we cannot know, right?
Precisely.
But he has another take on it.
To ask why is to elicit a response. Because in science, it seems to me that why X is the fundamental question to ask for. Its intent is to elicit the causal entailment structures underlying X. Without the causal structures, we have merely lists of data and phenomena, but no organizing principles or explanatory power. It is only with hypothesizing and elucidating the causal structures that we have organizing principles and explanatory power. That we have science. Imagine asking a physicist, why does the cannonball arc as it travels through the air rather than travel in a straight line? And the physicist responding, we don't ask why questions in physics. Or asking a geologist, why is the top of that hill rounded in that manner? And the geologist responding, we don't ask why questions in geology. Of course, those would be absurd conversations because those are perfectly valid questions. And there are perfectly valid answers that involve the causal structures involved on those scenarios. There can, of course, be unscientific answers to why questions. For example, the hill wants to be less angular, so it became more rounded. But such mistakes surely do not and cannot lead us to eviscerate why from the scientific enterprise. It seems that perhaps in the struggle to shed itself of an inherited legacy of vitalistic, anthropomorphic, teleological, and religious answers to why questions, biology has chosen a linguistic strategy to avoid such categories of answers by simply prohibiting questions of the form which may lead to such answers. It is perfectly valid to ask, why did organ X evolve in species Z? To deny this is to deny the role of causal entailments in biological nature. In effect, denial of these kinds of questions as being scientific leaves a void to be filled with nothing but vitalistic, anthropomorphic, teleological, and religious answers. Why or not we currently have in science sufficient data or robust answers to such questions is quite beside the point that the question is valid. If need be for palatability, such questions can be temporarily formulated as what questions? Why does the cannonball arc as it travels through the air rather than travel in a straight line? Can be rewritten as, what are the causal entailment structures that underlie the phenomenon of the cannonball arcing as it travels through the air rather than traveling in a straight line? Similarly, why did organ X evolve in species Z can be rewritten as, what are the causal entailment structures underlying the phenomenon of the evolution of organ X in species Z? Surely no trickster can deny the scientific validity of the point. Again, the tractability of this particular question is beside the point. Such what reformations leave us with an answer in the form of an enumerated list of causal entailment structures. This is by itself not enough. In this sense, what is more passive than why? Ultimately, we want to return to answering the why by taking those enumerated causal entailment structures and employing them in logical, mathematical models and leveraging the explanatory powers they can provide. Thus, it is that science is ultimately concerned with why questions and the answers they generate. Evading unscientific answers to questions about physical phenomena such as organisms by evading why questions would be, I think, quite destructive to science. Once again, thank you for the great podcasts that are so helpful, entertaining, and informative. Regards, Tim. Tim, that was, wow. You put a lot of time into that.
He certainly has.
So, before I met you, I'd never had a problem with why questions. Although I do agree with your... The point is you don't know why something exists. You want to know what its function is.
I have another word, though, that you could use. It's how.
Every use of why may not be illegal, but we try and avoid it just like we try and avoid anthropomorphizing viruses, just as a general way to avoid going down the road, right?
Sure.
So why does this fish have a fin that looks like this? It's a weird way to put it. You would like to know what is the function of the fin in this shape, right?
Bet. Yeah.
So it's all a matter of being more precise in your questioning.
That's right. So I like the word how as a substitute for why. That just allows you to work on the mechanism, that's all.
Mm-hmm.
What do you think, Daniel?
You know, my wife is really exacting with the children on their manners, and I try to. You know, so if they want a glass of milk, they don't say, "Can I have a glass of milk?" I say, "I don't know, can you?"
Yeah.
Are you able?
But may I? Would you please get me?
Sure.
And I was a bit of a stickler when the kids were younger. I was so worried that they wouldn't have a scientific approach to life. And I remember we're driving in the car, and one of my children asks, why do our baby teeth fall out? And my wife answered, to make room for the adult teeth. I was like, oh, I can't believe you said that. And and I think sometimes we can be a stickler. I don't know if it's that important whether or not we use the term why, is that we understand.
Right.
That in science, the answer is a how. And, the why questions are maybe metaphysical or theological.
Exactly.
And we're just restricting ourselves. And I think that's fine. But I realized I was a little obnoxious to my wife by demanding that every simple answer be couched in what is the evolutionary advantage of evolving to have two series of teeth.
So my son takes the can may thing a bit further. I'll say, Devin, can you take out the trash? He will say, it's may you take out the trash, Dad. No, come on. That's not right.
No.
May you take out he insists.
Right.
Robin sends us an article about if you can use extinct as a verb. We talked about this, didn't we?
Extincted, yes.
Did you say extincted?
I did, because I've seen it, and I know that some people are using it, and they're using it properly to suggest that the animal used to be, and now it isn't.
Yeah, I don't think you can use extinct.
Yeah, I think this link basically says no.
Yeah.
Yeah, so at least one source of usage says no, but that doesn't mean it's the definitive
It's
source.
a living language, and if we have enough listeners who adopt extinct as a verb, it's all over.
Yeah.
But he sent a link to a page, a conjugating page, Verbix, and they conjugate extincted. So, but that's just one of these auto forms. All right, our last one is from Mary.
Go ahead.
I didn't get to read one. Did you want to read Mary's?
No,
I don't care. It doesn't matter.
I'll read it if you'd like. So Mary writes, Dear TWiP, Part 1.
No.
Partway
No,
through.
part
Maybe Vincent should read this.
No, it's okay.
One.
Just read it, Dickson.
I'm trying.
Put your face on the mic.
You think I should move up closer to the microphone?
Well, just look at, like this.
I'm trying my best here.
Your best is not
Under
enough.
duress.
Under duress.
Okay, here we go. Number 1. Partway through episode 84, Bigfoot. I cannot help but wonder this. If the substance secreted by the filaria that inhibits division endothelial cells of the lymphatics, I read that directly, by the way,
It's fine.
mentioned by Dick, were isolated, identified, and its mechanisms determined, would we be looking at a possible application for cancer treatment? That sounds like a plowshare concept. I think it's worth looking into.
Isn't that what you tried to do with the endothelial factor of Trichonella?
No.
Angiogenic
factors. Yeah, that's the right approach. That's the hint that there may be something worth looking into. That's all. Number 2, my career goal is to become a registered diagnostic medical sonographer. I will be on the lookout for parasitic worms from heretofore. And in fact, for the case history that we just discussed and gave away, Paragonomus, you could have detected it like that. Number 3. This is my first TWiP episode, and I usually listen to TWiV during weekends at work. As a receptionist, I'm chained to a phone in a mini plexiglass fortress. Your podcasts are a delight while attending to bureaucratic paperwork. Number 4, can you suggest a social platform, Twitter, etc., to broadcast questions like item number 1? I have no problem exposing my scientific naivete to a public forum. It seems like a waste to have my curiosities disappear completely. Given a financial situation that prevents the education needed for a career with serious bench work, I want to throw my ideas to the wind and have the professionals sort it out. Though during retirement, I fully intend to take advantage of free community college classes offered to seniors. Keep up the great podcasts. You wonderful folks connect science to the public, and that is a noble service indeed.
I would suggest you use Twitter because it's a good way to ask questions, Mary. You could also consider Facebook. There's a huge community of people there. We have a TWiV page, facebook.com/thisweekinvirology.
Yep.
You can ask questions. There are lots of very knowledgeable scientists there who can help you out. And just follow certain people who seem to be knowledgeable about science, and that'll help your word get out as well. So I'd suggest Twitter and Facebook to start out. And that's pretty cool to find out who's listening. I always like to hear it.
It is.
And it's very cool that people with jobs like Mary's listen and try and enrich them. I think this is great. So this is we're doing good stuff, Dickson.
We are, even though sometimes we're wrong.
Even though you thought this was all a waste of time, right, Dickson?
I never thought this was a waste of time.
You did.
Ever.
You did.
Never. You just step outside
for
and say
years,
that.
For years. Only now have you come around, witnessed
Vincent,
by the fact that you
Vincent,
asked Daniel to join us. What?
I'm a big fan.
You don't have to be a fan. You just have to believe.
I'm a big fan of doing this.
All right, good.
No, hopefully we are doing this. We're educating. We're getting some more knowledge out there.
And you can do it in a lighthearted way, too. I mean, you don't have to be a heavy-handed,
Yeah, that's why I pick on you.
dogmatic person that's always right. We're never always right. No one's
Of course
ever
not.
always
We never
right.
said we were
Nobody.
always right. No one is.
That's right.
Even the lawyers and
Exactly.
science writers, even the doctors aren't always right, Daniel?
Well, I
Heavens
think we
forbid.
talked about that. The height of your arrogance is the last day of medical school, and then hopefully, if you're paying attention, that should drop to a very low level, and you should be incredibly humble the first day you start trying to take care of patients and realizing how complicated it is and how often you don't know the answer.
What I think is a really important part to realize that the patient is looking to you to help them.
They are.
And they're helpless. They have no clue most of the time, and they want you to help them. And many physicians are really insensitive to that, unfortunately. It's hard to teach, I suppose, but you have to. These are people you're taking care of.
Yeah.
Treat them like people. Of
Yeah,
course,
the
to
best-
some people that's not good.
The best doctors I've encountered are good listeners. That's what it takes, a good listener to wait
hopefully-
for the story and then say something.
Yeah, hopefully people are realizing that our cases, you get the answer not by a CAT scan or an MRI or something expensive, but by spending time and asking the right
Correct.
questions. If you listen long enough and ask the right questions, the patients will tell you everything you need to know.
You got to be curious.
And then you confirm it.
Right.
Yeah, that's the cheapest diagnostic, right? As
Yes,
you
I think
said
we
last
talked
time.
about
It
the
is. No, it is.
cheapest
It
thing you
is.
can do.
That's
I like
right.
that
That's
very
right.
much.
That's right.
Well, that is TWiP 85, gentlemen and listeners and ladies. You can find this and all the others at microbeworld.org/TWiP. You can also find us on iTunes or on your favorite podcatcher. And there are all sorts of them out there. And do send us your questions and comments to TWiP at twiv.tv. That's TWIP at twiv.tv. And you can send us your thoughts about the next case study or any other questions that you might have. Dickson Despommier, who is one of the TWiPumvirate, is right here
That's right.
in the office at Columbia University. I get to see him every day, which
You
is
do.
kind of sad, but on the other hand,
The feeling is mutual. However you want to say that, I agree entirely with you.
if I didn't see you every day, it would be hard to build the rapport that we have.
Vincent, you
built.
don't see me every day. Sometimes you accuse me of going on vacations or going to trips or trivialities of...
Yeah, Dickson, it seems like I see you every day.
it just seems that way. That's
You can
right.
find Dickson at verticalfarm.com.
You can.
Thank you, Dickson.
You're very welcome, Vincent.
I also see Daniel every day. He doesn't bother me as much as you
He
do.
sits in a different place. That's why.
Yeah, his
Otherwise,
office
he'd
is
bother
far
both
away.
of us. That's not
Daniel
true,
is right
but
here
in a
at
good
Columbia
way.
University, where he takes care of patients and does research. Thank you, Daniel.
Oh, a pleasure again.
So we got a good chemistry, they say, huh?
I think that's true, by the way.
Yeah. Which it's thanks to you because you picked Daniel. I didn't know him really.
Well, come on. I didn't make
You know why?
him
Because you
happen.
sit
He was
out
already
there and chat with anyone who walks by.
I try. I
that true,
desperately
Daniel?
try to
Pretty much.
occupy my life.
Dickson is an extrovert. He is a friendly, chatty fellow.
So when you first met him, did you walk by and he said hello to you, or did you say hello to him?
That's a good question, actually.
I believe Martine introduced us. Is
Yeah.
that true?
Yeah. I mean, you came in for some coffee or something like that.
Yeah, I came in to grab coffee.
But then we
or-
traveled together down to the American Society for Tropical Medicine and Hygiene meeting.
Why did you go to that, Dickson?
I went there to meet up with the other authors of
Oh,
our
your book.
textbook in an attempt to revive interest in making a 6th edition.
Actually, that was a great trip for me. I ended up connecting with a researcher, and then through him a bunch of researchers at the University of Malawi. And hopefully, if I can get through this IRB, which I'm hoping will happen this week, we're going to be looking at influenza-like illnesses in Malawi, in Blantyre, trying to come up with a clinical prediction tool, as we say, a way of figuring out what diagnosis they might have and what treatment is appropriate without having to resort to all these expensive tests where they really don't have access to those in a place like Blantyre, Malawi.
So your book is not going forward, right?
Not yet.
Not yet. Not ever.
No, it's not. It's not yet. It is available through a PDF. I would gladly send a copy to anybody who writes me.
I'm Vincent Racaniello. You can find me at virology.ws. The music you hear on TWiP is composed and performed by Ronald Jenkins. You can find his work at ronaldjenkins.com. You've been listening to This Week in Parasitism. Thanks for joining us. We'll be back soon. Another TWiP
is parasitic.